Let's watch the explanation.
It's all part of the miracle of cloning.
Hello? Hello, John. Law extraction has never recreated an intact DNA strand.
Not without master sequence gaps.
Where do you get 100-million-year-old dinosaur blood?
Shh! Just one drop of your blood contains billions of strands of DNA. A DNA strand, like me, is a blueprint for building a living thing. 100 million years ago, there were mosquitoes, just like today. Sometimes, after biting a dinosaur and get stuck in the sap, the tree sap would get hard and become fossilized, preserving the mosquito inside. Jurassic Park scientists came along. They extract the preserved blood from the mosquito and dino DNA. It's full of holes. And supercomputers break down the strand in minutes. Virtual reality displays show our geneticists the gaps in the DNA sequence. We use the complete DNA of a frog to fill in the holes and complete the code.
So my first question for this is, when I was a kid, I was like, this is awesome. This is totally possible. Is it possible? It feels possible-ish.
I think so. I think it's possible. I think it's CRISPR, pretty much.
Oh, really?
Mm-hmm.
What about this? Wouldn't the DNA degrade over lots of millions of years? So, you'd only have these tiny segments, maybe.
Yeah, great point. I don't know. So I guess the question, we could reframe it as if we were to take a drop of blood, could we dry it out, crush it up, powderize it, put liquid back into it, and then do DNA stuff, centrifuge it, and be like, we can recombine it? Or does it need to be in a liquid state? Or does the act of crushing it, freezing it, turning it into an amber inside of a mosquito, does that destroy it?
My instinct is saying that maybe each process along the way might be okay, but you got 65 to 100 million years of time elapsed. That's a lot of time for stuff to degrade.
And you have nothing to check it against.
And you have nothing to check it. If you had a pure sample to check it against, then that would feel doable. But I don't know. It seems maybe possible, but I could also see it being this is just a mess of broken up DNA that we're just not going to be able to do anything with.
I'm not a geneticist. I'm just a dreamer. Figure out how to do it.
Yeah. And then there's also, I didn't realize, augmented reality.
Oh, very cool. So this was 1993 when this was filmed, and AR, augmented reality, virtual reality was starting to come up online, but this was super visionary. The thought about this a long time ago.
Right. And then, so I guess this is the extraction process. It looks a little imprecise to me.
Of course.
Yeah. Well, I want to get in there with something. Okay, it's a super fine needle, but I think it'd be even sharper.
Absolutely.
Mm-hmm. There's probably not much DNA in there, if there is any. Yeah.
I mean, if you knew the gene sequence of that mosquito, you could just blend the whole thing into a slurry, and then whatever is the mosquitoes, just subtract it. I mean, if that's even possible, if you can do that.
Because it's also a prehistoric mosquito,
right?
Which is no guarantee there's a corresponding modern mosquito, because they've also evolved.
That's right. Absolutely.
Yeah.
I don't know. If you're a geneticist, tell us how accurate was Jurassic Park.
Yeah. Is it maybe someday with super, super good technology possible, or is it just not going to happen? Seems legitimate to me.
Seems plausible to me. This is a cool scene. So part of the tour is seeing the scientists working, and so it's cool for the tourists. They get to see what's happening behind the scenes. But this to me feels like working in a fish tank. This feels to me like I have tourists constantly monitoring me. Super uncomfortable.
I guess I have seen this in actual museums where scientists actually have lab equipment behind a fishbowl glass thing.
That's right. Was that Chicago we saw that?
We saw it in Chicago, yeah.
Again, that was pretty cool.
I wonder if that helps funding. Show it to the public, get them excited. True. Funding is flowing.
It feels more legitimate as opposed to some egghead somewhere said some number. No, you could actually see what they're doing.
Right.
And I guess, I get it. From the scientist perspective, it's people finally get to see me doing my thing and appreciate. Okay, I see. This is actually pretty good.
It's been pretty good. Plus, it is an amusement park, a zoo. It's Jurassic Park.
Yeah.
And so people would be like more than curious as to what the heck, how this is done, because it's so incredible.
I mean, it's effectively magic.
Yeah. Dr. Grant asked this weird question. We were trying to figure out why.
Wait a minute. How do you interrupt the cellular mitosis?
Can't we see the unfertilized eggs?
Softly.
So why would you want to interrupt cellular mitosis?
I don't know. So first question I have is, what is mitosis? What's mitosis versus meiosis?
Yeah, so let's look that up. I think mitosis is when a cell splits in two and has identical DNA in the nucleus.
And versus meiosis?
Yeah, so let's zoom in here. So mitosis produces two genetically identical daughter cells from a single parent cell, whereas meiosis produces cells that are genetically unique from the parent and contain only half as much DNA. So this is like when a sperm and an egg meet, they combine to make a whole new set of DNA that's got half from mother, half from the father. Is that right?
I see. So haploids combining to make a diploid?
Haploid. I was going to use the term zygote. Is that right? Haploid? Haploid. Let's go to ploidy.
Okay.
Ploidy is the number of complete sets of chromosomes in a cell, and hence the number of possible alleles for autosomal and pseudo-autosomal genes. Okay, so we want a diploid. Somatic cells, tissues, and individual organisms can be described according to the number of sets of chromosomes present, the ploidy level: monoploid 1 set, diploid 2 sets, triploid 3 sets, and so on.
So humans are diploid type of genes, and so a haploid would be a sperm and another haploid would be an egg?
What's haploid would be?
Half of 2 is 1?
So that would be a monoploid?
I guess so. So my understanding of this is there are different types of DNA pairings, and some species, like humans, have diploid sets of genes. Other things, I think bananas are triploids. They have like 3 chemical something in their DNA groupings. This is exceeding my high school biology.
This is exceeding us. So the question was, why does he want to interrupt cellular mitosis to do this? You'd think you'd want to interrupt cellular meiosis, because that's when the genes come together in sexual reproduction.
Right. So you can alter those set of genes and then let mitosis go on and let it replicate. Fine.
Maybe mitosis is the better time to do it, to do the manipulation.
Right. So on one hand, if you do mitosis across a single cell, you're only done mitosis across that one cell, and so all the other ones are replicating the original genes, which is not what you want. But I guess you also, if you interrupt mitosis, if you're able to separate one cell, then you have like the full set of genes, the full combination of whatever it's going to do. And so then when you alter that gene and you put in the new gene, then it's like, here's your whole thing that you're going to do, go do it. I'm not a geneticist. I have a high level understanding of CRISPR, but I imagine if I were to do changes prior to the mitosis, prior to the final set of DNA, then if I take 2 haploids, which I've altered, if I combine them, will they combine in the way that I anticipated? Maybe not. So in that way, then perhaps mitosis is the way you want to break it apart.
And so he does say interrupt mitosis.
Interrupt the cellular mitosis.
Let's see here one more time.
How do you interrupt the cellular mitosis?
So you interrupt the mitosis and then put in what you want, and then let it continue.
I guess so.
That sounds super advanced.
That's what I learned from Jurassic Park.
Yep.
Jurassic Park and 2 Wikipedia pages.
That's right. That's what we learned. He was on that question.
He's on it. Yeah, he knows. Wait, he's a paleontologist. He knows a lot about cellular mitosis.
Does he know a lot about cellular mitosis?
He knew where to interrupt it.
He knew where to interrupt. That seems like an advanced question, doesn't it?
Yeah.
Where it's like, how do you do this tech, this Jurassic Park tech? Mitosis is the place.
Maybe his favorite reading material is just scientific journals all the time.
Oh, well, possibly.
Watch this classic scene.
How do you know they're all female? Does somebody go out in the park, pull up the dinosaur skirts?
We control their chromosomes. All vertebrate embryos are inherently female anyway. They just require an extra hormone given at the right developmental stage to make them male. We simply deny them that.
Life will not be contained. It crashes through barriers.
You're implying that a group composed entirely of female animals will breed?
No, I'm simply saying that life finds a way.
I mean, it's classic.
I found a way.
He ends up being right, although I'm not sure he'd be right all the time.
That's right.
And plus, at the beginning, this guy's doing some good work.
Oh, yeah.
And Ian Malcolm comes in with some ridicule language.
You know they're all female. Has somebody go out in the park, pull up the dinosaur skirts?
We control their chromosomes. All vertebrate embryos are inherently female.
Is that some insecurity on Ian Malcolm's part?
Totally, yeah.
He's like, no, I'm the leather jacket guy. F this guy.
I'm the math guy. I'm the smart. I make the statements, but it's like the better explanation has got the scientist that actually does it.
That's right. But and then Ian Malcolm ends up being right.
He's actually right. He's actually exactly.
They just require an extra hormone given at the right developmental stage to make them male. We simply deny them that.
Life will not be contained. It crashes through barriers.
You're implying that a group composed entirely of female animals will breed?
No, I'm simply saying that life finds a way.
Finds a way. Wouldn't it be better to have all of the dinosaurs be male?
Well, how do you figure?
Well, if you have a whole bunch of females and one dinosaur somehow turns to male, you've now got a bunch of pregnant dinosaurs. Whereas if you have a bunch of males and one turns female, you probably have a huge fight, and then you have one pregnant dinosaur.
That's right. So it sounds like the failure mode for if the dinosaurs can change sexes, then mostly dudes and one woman. That seems safe.
That's going to control the population and let you get ahead of the problem before it balloons.
Mm.
Whereas if it's a bunch of females, I mean, everyone's getting pregnant.
Just exponential growth right away.
That's right. I mean, immediate.
Instantly.
Right. Especially if one female can have like 5 to 10 eggs.
That's right.
Whew.
It's a fractal.
It's over. It's a fractal.
Yeah. Fractal diagram is like 1 makes 5. Those 5 make 5. These 5 make,
I see. Yeah, so interesting they chose to make them female instead of male. I guess it's that mechanism that he was talking about. It's just everything's female unless you give them that
hormone. But then just give them all the hormone.
Just give them all the little hormone.
Yeah.
You got a bunch
I assume it's synthetic hormone. They're not like extracting it out of cows.
Right. Yeah, that's true. Yeah. So I think it's better to make them all male. But
get them all some dino Xboxes, a bunch of male dinos on the island hanging out, chilling. Like
that's right.
Yeah.
An apartment with nothing in it. It's perfect. They don't even need it.
It's like a lawn chair and like a beer cooler.
That's right.
The dino size.
Why are the dinos so chill? They don't need anything.